When we talk about health today, we often talk about parts, the gut, the brain, the immune system, as if they are separate departments of the same body. But the more we learn, the clearer it becomes that the human body, it doesn't work in parts. It's a system and a network of constant communication. And the gut sits right at the center of that network. When most people think of the gut, they think of digestion. Food goes in and nutrients get absorbed, and then waste comes out. And that is true, but it's only the surface layer of what's really happening. The gut is our largest immune organ, housing about 70 to 80% of our immune cells. And it is also home to trillions of bacteria, which is what makes our gut microbiome, which forms an entire ecosystem living inside of us. And here is what's extraordinary. These microbes, they don't just digest foods, they also talk to the body. And they do it through chemistry, through the molecules they produce as they break down what we eat. And these molecules are called metabolites, and they move beyond the gut wall and send messages that affects our metabolism, our immune system, and even the brain. So the gut isn't just a digestive tube, it is a communication hub, a central interface where the outside world, everything that we eat, breathe, and feel is translated into signals that guide our inside world, our physiology, our mood, our energy, and our resilience. And over the past 15 to 20 years, new DNA technologies have given us an incredible gift, which is that we could map which microbes live inside of us. And that led to an explosion in microbiome research and also a new way of thinking about health. And then for a while, the question was mainly who is there? Which bacteria are found in people who are lean versus people with obesity? Which ones are more common in those with gut disorders or in those who stay healthy as they age, for instance? And those questions are exciting, but they've also revealed a puzzle, which is that two people can have nearly the same microbiome composition, yet sometimes completely different health outcomes. So the next question became not just who is there, but what are they doing? And that is where metabolomics changed the game. Instead of looking at microbes as names on a list, metabolomics has allowed us to look at what they produce, the chemical fingerprints of their activity. And it lets us see the function of the microbiome, how those trillions of bacteria are actually influencing our biology. And in a sense, these metabolites, they are the language of the gut. They are how our microbes communicate with us. And when that language shifts, when certain messages go silent and other ones become louder, it can change how our whole system behaves. So now when we look at this communication system from a distance, we can see how profoundly interconnected it is. The gut talks to the immune system every second. The immune system feeds back signals that influence microbial activity. And the brain listens to all of it through the vagus nerve and through the molecules that circulates in the blood. So the gut is not a standalone organ. It is a systems organ and a biological crossroad where immune, metabolic, and neural networks are constantly communicating. So when this network is balanced, we see stability across multiple systems. We see steady energy, lower inflammation, and better mood regulation. But when it is disrupted, that imbalance echoes throughout the body, in the liver, in the brain, and even in how we age. And this is where tryptophan becomes such an elegant example of systems biology in action. Because tryptophan is an essential amino acid, which means that we have to get it from food. But what happens to it after that depends on the conversation between our cells and our microbes. And we can think of tryptophan as standing at a crossroads with three roads ahead. And the first is the kynurenine pathway, which is the main route handling around 90 to 95% of tryptophan. And this pathway is essential as it produces NAD+, which is a molecule that every cell needs for energy. But it is also the body's stress response. So when inflammation or cortisol levels rise, enzymes like IDO1 and TDO will open the gates wider, which pulls more tryptophan into kynurenine. And if this stays active for too long, it creates a biochemical environment that fuels oxidative stress and immune strain. The second one is the serotonin pathway, which is a smaller road with just about 1 to 2% of traffic. And here, tryptophan becomes serotonin, which affects mood, sleep, and gut movement. And then we have the third road, which is the one that interests us the most, the microbial indole pathway. And this is where gut bacteria, they take tryptophan and turn it into compounds called indole derivatives, including one unique one called indole propionic acid, or IPA. And these are not just byproducts, they are signals of cooperation. When this microbial pathway is active, it tells us that the gut environment is healthy, that the right microbes are being fed, that inflammation is low, and that the body and the microbiome are in balance. So in other words, when the system is under stress, tryptophan will flow into kynurenine more heavily, but when the system is balanced and nourished, it flows more into the indoles like IPA. So that shift from kynurenine to indole metabolism is one of the clearest biochemical signatures of resilience that we can measure. And I like to use this traffic light metaphor where we can think of tryptophan as arriving at a busy traffic intersection. And then we know that diet and microbiome diversity will turn the green light on for the protective indole road, whereas stress and inflammation turns it green for the kynurenine highway. Now we can't change the map, but we can influence and change the traffic. Now let's go more into the details of this unique marker, because IPA is one of the most fascinating compounds discovered in recent microbiome science. And it is made exclusively by certain gut bacteria when they have the right conditions, particularly when they are fed dietary fiber. And its effects reaches far beyond the gut. IPA, it strengthens the intestinal barrier, which is the thin, selectively permeable layer that keeps unwanted molecules out and nutrients in. It also calms immune cells that would otherwise stay in overdrive. And it acts as a powerful antioxidant, protecting cells from oxidative stress. And it even influences metabolism by helping to regulate blood sugar. And what's remarkable about IPA is that it doesn't just reflect a healthy gut, it helps maintain it. It creates a feedback loop. The more IPA your microbes produce, the stronger the barrier becomes, and then the more stable the system stays. So you can think of IPA as a guardian molecule, a molecular signal of cooperation between us and our microbes. And diet is one of the strongest determinants of whether tryptophan flows into the kynurenine or the indole road. And fiber is the single most important driver. Our ancestors used to eat well over 100 grams of fiber a day, but today most people in industrialized countries they get less than 20. So that shift has completely reshaped our microbiome and with it our metabolite balance. When we eat a low fiber, high fat Western diet, microbes lose their preferred fuel source and they start breaking down mucus and protein residues instead, which creates more pro-inflammatory byproducts. And at the same time, immune activity will rise, which pushes tryptophan more heavily into kynurenine. But when we get enough fiber, the opposite happens. Microbes will ferment fiber into short-chain fatty acids like butyrate, which helps to suppress inflammatory signals and IDO1 activity. And this then frees up tryptophan to flow into the microbial indole route, producing more IPA and fewer inflammatory metabolites. And fiber doesn't act in isolation. It also interacts with fat quality. So, for instance, our omega-6 to 3 balance will influence this. And it also interacts with polyphenols, which acts like microbial selectors, enriching species that produce beneficial metabolites. And of course, also with lifestyle factors such as stress, sleep, and physical activity, all of which influence immune tone and the enzymes that govern tryptophan metabolism. So health in this light isn't about a single nutrient, it's about coherence and how our diet and lifestyle work together to shape this network of signals. Now, to understand what really shapes IPA production, I want us to look a little closer on its biggest driver, which is fiber. And this figure here shows how fiber is a game changer for tryptophan metabolism through the proposed mechanisms of how it influences it. First, fiber slows down absorption, so then more tryptophan reaches the colon. And then second, when fiber is fermented, we know it produces this short-chain fatty acid butyrate, which can down-regulate IDO1 expression and inflammatory tone, which means then that fewer tryptophan molecules are drained into kynurenine. And then third, fiber feeds the beneficial microbes. So with fiber, we get less tryptophan lost to the stress road and more directed into the protective indole road. And this is why fiber is such a powerful driver of resilience. And population cohorts and intervention studies indicate the same thing. With higher fiber intake, we see higher circulating IPA and more favorable tryptophan ratios. And this is not just in theory, so I want to look more closely into what this balance means in real-world conditions from the scientific literature, starting in the liver. Because in the liver, this balance makes a clear difference. And as you can see on the figure here, in a healthy state, certain gut bacteria will convert tryptophan into indole compounds like IPA. And these IPA can activate a receptor called the aryl hydrocarbon receptor, which is a molecular sensor in the gut lining that detects the signal from the microbes and helps keep the immune system balanced. And when the aryl hydrocarbon receptor is activated by IPA, it strengthens the gut barrier by promoting the renewal of intestinal cells and the production of tight junction proteins that seals the gut wall. And it also calms immune activity and helps suppress inflammation. So the result is fewer endotoxins leaking into the bloodstream, less liver inflammation, and improved fat and energy metabolism. But in metabolic liver disease, when the gut microbiome loses its diversity and indole production drops, the opposite happens. The barrier weakens, bacterial toxins leak through, and inflammation is switched on in the liver through pathways like the NF-kappa beta, which leads to fat accumulation and tissue damage. And in the gut itself, the contrast is just as striking. When the microbiota is balanced, tryptophan is converted into indole compounds like IPA, which, like I said, can activate the aryl hydrocarbon receptor in the gut lining, which strengthens the gut barrier and promotes immune balance and keeps inflammation under control. But in inflammatory bowel disease, when dysbiosis sets in, the microbes that produce these beneficial indoles are reduced, and as a result, more tryptophan is diverted into the kidney pathway, which is driven by inflammation. So the gut barrier then becomes leaky, immune signals turn pro-inflammatory, and chronic inflammation takes hold, which also fuels a vicious cycle that will help maintain disease activity. And the same principle apply as we age. Over time, the gut microbiome often loses its diversity, meaning that fewer beneficial bacteria and a drop in protective metabolites like IPA will happen. And with less IPA, the gut barrier weakens, immune balance shifts, and low-grade chronic inflammation becomes more common in a process often called inflammation. But not all aging follows this path. Studies of healthy centenarians show something remarkable, which is that they often maintain a diverse, balanced microbiome, and they continue to produce indole metabolites, and they preserve strong barrier and immune function. So, in other words, aging is not an inevitable decline necessarily. It is deeply influenced by the state of our microbiome and the molecules it produces. The same imbalances that affect our gut and metabolism also reach the brain through what is known as the gut-brain axis. And when the system is imbalanced, the microbial indoles like IPA helps to protect the blood-brain barrier, reduce inflammation, and support the production of key neurotransmitters such as serotonin and dopamine. But when the gut is imbalanced and the barrier becomes leaky, inflammatory signals rise again and kidney pathway metabolites will build up, which is a process that can tip the brain towards stress, mood disorders, and cognitive decline. And this is also why gut health is increasingly being linked to conditions such as depression and neurodegeneration. So across all these systems, the liver, the gut, the brain, and even aging, we see the same pattern emerges. When tryptophan is channeled into microbial indoles like IPA, we see protection. We see stronger barriers, calmer immune responses, and greater overall resilience. When more tryptophan is diverted into the kidney pathway, we see the opposite: weakened barriers, higher inflammation, and greater vulnerability to stress and disease. So the key takeaway I want you to take from this is that this balance isn't fixed. It is something that we can influence. And diet, especially fiber intake, as well as sleep, exercise, and of course microbial diversity, all shape how this pathway flows. So in other words, the way that we live and eat, it doesn't just feed us. It steers the chemistry that connects our gut, our metabolism, and even our mind. And by resilience, we mean tighter barriers, lower baseline activation, and steadier metabolic control, which are measurable hallmarks of a system that adapts and recovers efficiently. So, now that we know that this balance between indoles and kidneys isn't fixed, it's something that we can shape through what we eat, how we move, how we sleep, and how we manage stress. The next question is, how do we actually see it? How do we measure whether our gut and metabolism are moving towards resilience or drifting towards strain? So this is where measurement becomes powerful because for decades we've been able to see the microbiome, but not necessarily directly its function. We could catalog bacteria, but we couldn't easily measure what they were producing or how those molecules affected us, and especially not in a simple at-home tool that is affordable. But now we can with the gut health tests. And by measuring three key metabolites in blood, which is tryptophan, kynurenine, and IPA, we can get a more functional snapshot of how this system is performing. So we have tryptophan, which shows the available substrates or the fuel at the crossroads. And then we have kynurenine, which reflects immune activation and inflammatory tone. And we have IPA, which reflects the microbial health and cooperation. And from these, we can derive ratios that act as system indicators. The kynurenine to tryptophan ratio, which is our immune stress indicator. The IPA to tryptophan ratio, showing microbial efficiency, and thus is our gut microbiome efficiency marker. And then we have the IPA to kynurenine ratio, which shows the overall balance between protection and stress in our microbiome immune support marker. And when we integrate them, we get the gut health index, which functions as a snapshot of whether your system is leaning towards resilience or strain. And I just want to stress that this is not a diagnostic test. It doesn't label diseases, but instead it provides actionable insights into how your gut and metabolism are working together right now and how diet and lifestyle can shift that balance. So this is what we measure with the gut health tests. And in order to define meaningful cutoffs in these tests, we haven't turned to disease thresholds. But we've looked at two groups in our internal data sets that represent the opposite ends of the dietary spectrum. We have those with low fiber intake and low omega-3, typically with higher inflammation and lower microbial activity, and those with high fiber intake and high omega-3, typically with lower inflammation and stronger microbial function. And across these groups, the pattern is striking. We see higher IPA, we see higher IPA to kynurenine and higher IPA to tryptophan ratios in the high fiber, high omega-3 group, and lower kynurenine to tryptophan, meaning less immune stress. And when we compare those trends to what's published in the scientific literature, we see the same directionality. Higher dietary fiber and omega-3 intake consistently associated with higher IPA and lower kynurenine to tryptophan. So our reference zones, low, moderate, medium, high, and high, they aren't arbitrary. They are grounded in both our internal population and the wider body of scientific evidence. But they don't diagnose disease, they describe direction, where the system is leaning towards resilience or towards strain. And here you can see an example from the test report and how it will appear to the user. And again, I want to emphasize that these zones are directional indicators, and they show you where your biology sits along the continuum from strain to resilience. So this approach, it keeps the interpretation firmly anchored in preventive health, reflecting biological shifts observed in healthy populations and not clinical thresholds. So in other words, the results guide an understanding and action and not classification. So what this really represents is systems thinking in practice. We're not measuring a single molecule, we're measuring a pattern, the interplay between the microbiome, the immune system, and metabolism. And each molecule is like a data point, but when we put them together, they form a picture, a system's fingerprint of how your body is functioning. And that's what makes this so exciting. It's not about replacing medical diagnostics. It's about expanding our understanding of preventive health and about seeing biology as a dialogue that we can influence through diet and lifestyle. So to summarize, when tryptophan flows into microbial indos like IPA, we see cooperation, we see balance and resilience. But when it flows too much into kynurenine, we see strain, inflammation, and reduced adaptability. And because these molecules circulate in the blood, we can now read that balance directly, turning the invisible chemistry of the gut into a measurable window on systemic health. And that's the promise of this approach. It's not just about the microbiome, it's about what it's saying and how we respond. So the gut health test gives us the first practical way to listen, to translate the gut's molecular language into something personal, actionable, and deeply human. And that's also what makes the gut health test the next generation of gut testing, translating this cutting edge science into clear and personal and actionable insights. It bridges the gap between research and real life, which gives individuals a way to understand how their choices shape their biology. And in the bigger picture, it transforms complexity into clarity, from thousands of microbial names to a few measurable molecules that reveal whether your system is trending towards protection or stress. And so again, this is systems thinking brought to life, turning those complex biological networks into something personal and actionable. And because when you can see how your whole system responds to what you eat, how you sleep, and how you live, you can also start steering it towards resilience. So what makes this so powerful is that it represents a shift from observing disease to understanding health. And instead of waiting for something to go wrong, we can now measure how well our system is adapting, how balanced the dialogue between the gut, immune system, and metabolism really is. And this is the essence of preventive health, not prediction of disease, but detection of imbalance while it's still reversible. And the gut health test gives us a way to see whether the body's internal chemistry is trending towards protection or strain, and more importantly, help guide how to shift it back. Because when we can see change, we can also act on it. And that's how science becomes prevention and prevention becomes empowerment. Thank you.